So here we have an overall depiction of how these reactions correlate with each other. So we have glycolysis occurring in the cytosol. So it's important to understand why oxygen becomes so important. So oxygen is considered in these aerobic pathways to be the final acceptor of electrons. When it accepts these low energy electrons from the electron transport chain, once again, they're going to be carried in the form of hydride ions, and they're going to be used to eventually create that water molecule. Oxygen and hydrogen will create water. So what happens if we don't have oxygen present? If we don't have oxygen to accept these low energy electrons, everything gets backed up, meaning that the electron transport chain can no longer occur, because if it can't keep passing those electrons through, it's going to eventually stop. If you don't have an electron transport chain, that means that you're not creating that proton gradient. So you're not going to have those protons being pumped into the intermembrane space. If you don't have a gradient, you don't have ATP production, because ATP synthase makes that ATP by taking advantage of that proton gradient. So when the electron transport chain stops, that means that all of your NADH and FADH2 that have these high energy electrons, if there's no electron transport chain, they have nowhere to pass these high energy electrons. So that means that these electron carriers were built up in the cell. That means that there's not going to be very much free NAD plus or FADH plus. So that means that the Krebs cycle will stop too. Why are you generating more electron carriers when you already have too many? So the Krebs cycle shuts down. So basically, if you don't have any oxygen, this disrupts the flow of electrons and will shut down everything in your aerobic pathways. So this brings us to our anaerobic pathways. How do we overcome this problem of oxygen deprivation? When there's no oxygen around, cells can still use glycolysis to make some ATP from glucose, because we know that there's a little bit of ATP that is generated from glycolysis. We also know that glycolysis is considered to be an anaerobic step. You don't need oxygen present for glycolysis to occur. But the important purpose of glycolysis is to regenerate NAD+. So overall, your glycolysis in this case in an anaerobic pathways is to regenerate NAD+, not to make a little bit of ATP. So oxygen, remember, is that final acceptor of that low energy electrons. And if you don't have oxygen present, everything else is going to back up and everything is going to shut down. So fermentation can occur to overcome this deprivation. So anaerobic pathways involve glycolysis, because remember, glycolysis is an anaerobic step, in addition to your fermentation pathways. Remember that the overall goal of these pathways of anaerobic respiration is to regenerate NAD+. This will help to keep those pathways going until oxygen becomes available, and then you can utilize those aerobic pathways to make the most ATP possible. So there's two different types of anaerobic pathways that we can see: alcoholic fermentation and lactic acid fermentation. Alcoholic fermentation is present in yeast cells, and this is where pyruvate can be converted to ethanol and carbon dioxide. So it's a very advantageous process for making alcohol. This is how alcohol is actually made, by taking advantage of the anaerobic pathways present in yeast. We can also have lactic acid fermentation. This is present in animal cells. Obviously, our cells cannot produce alcohol as a byproduct of fermentation. We produce lactic acid instead. In this pathway, we're going to convert pyruvate to lactic acid. So let's take a look at what is involved in these two processes. Once again, I want to highlight the goal of these processes is to regenerate NAD+. This will keep glycolysis going. This will keep a little bit of ATP being made until aerobic pathways can then be present. This diagram here illustrates those two processes: alcoholic fermentation in yeast versus lactic acid fermentation in animal cells. Keep in mind, once again, I don't want you to memorize these structures. However, I want you to be able to understand what is happening to the carbons. So once again, in glucose, we have glucose in both processes undergoing glycolysis. In this phase, we're going to convert that 6-carbon molecule of glucose into 2 pyruvate molecules. Those pyruvate molecules have 3 carbons each. In that process of glycolysis, we will generate 2 ATP molecules, and we will also generate NADH. Now, if oxygen is no longer present, this is when these pathways will take over. In alcoholic fermentation, we will have pyruvate being converted into a 2-carbon molecule, acetaldehyde. Remember, if we're going from a 3-carbon to a 2-carbon, we would then have the production of carbon dioxide, which is what we see here in alcohol fermentation. That acetaldehyde will then be converted into ethanol. Ethanol is a 2-carbon molecule. As acetaldehyde is being converted to ethanol, this is where we have the opportunity to regenerate NAD+. So it will take that NADH molecule and it will oxidize it to the NAD+ form. In lactic acid fermentation, we once again have glycolysis. We have the formation of 2 pyruvate molecules from 1 glucose. Except in this pathway here, we're going to take those pyruvate molecules and we are going to convert them into lactic acid or lactate. Once again, we have 2 molecules of lactate being produced. Keep in mind here, we're going from pyruvate, a 3-carbon molecule, to lactate, which is also a 3-carbon molecule. Since we haven't lost any carbons in this process, we don't see carbon dioxide being produced. Here we simply have the regeneration of NAD+. So NAD+ in both of these cases, lactic acid and alcohol fermentation, NAD+ will be used to keep glycolysis going. This will allow for the cells to break down glucose so it doesn't accumulate in the cell, produce pyruvate molecules, generate a little bit of ATP to keep the cell going. Once again, when oxygen becomes present, we now have pyruvate available that can easily be shuttled into aerobic pathways. And finally, a last important concept to note is that we have focused on the breakdown of sugar, specifically glucose molecules, and tracing it through these processes. But keep in mind, there are many other molecules that can be used for energy in the cell. Proteins can be broken down into their amino acid components. They can be shuttled into different processes of the cell. They can be shuttled into the end of glycolysis, they can be shuttled into the shunt step, and they can also be shuttled into the citric acid cycle. Various types of carbohydrates can be broken down into sugars. They can also be broken down into any of the byproducts or intermediary compounds within the glycolysis pathway. We can also utilize Fats, we can break them down into their constituents, the glycerol and fatty acids, and those can then be broken down into different components. So glycerol can be easily converted to G3P to enter into glycolysis, and fatty acids can easily be broken down into 2-carbon molecules to then enter into the citric acid cycle. And that will end the lecture on respiration.